So for this last sessions, I will present you one of the clinical applications using TVB. And I will present you with epilepsy. So just some introductory slides. So an epileptic seizure, it's a brief episode of signs and or symptoms. For example, when you see people having convulsions or losing consciousness, or altered behaviors that is due to an abnormal electrical discharges in the brain. So here you have two figures. One, it is a seizure recorded in the hippocampus of a mouse, and here in the human. So the seizures are characterized by an onset, then a various alternation of fast discharges, so with the hashtag, and of spike wave events with the stars, and an offset. These two generic patterns, so the low fast discharges and the spike and wave events, can be observed across species, like here in mouse and human, but also across brain regions. So if an epileptic seizure, it's a single event. The epilepsy, it's the recurrence of this spontaneous or unprovoked epileptic seizures. So 1% of the world population has epilepsy. 1/3 of this 1% is resistant to medications, and their only hope for relief is the epilepsy surgery. The pre-surgery evaluations consist in EEG, there's also MEG or fMRI. What we are doing in Marseille, it is the SEEG, stereotactic electroencephalography, where we have this type of needles that are inserted inside the brain. So the goal of the surgery is to find out where is the epileptogenic zone, so where the seizures start in the brain. And this is the target of the surgery. The problem is, one of the problems is even the patients, even if the clinician has identified the epileptogenic zone, after surgery, there's still a lot of patients who still have some seizures. And this level did not improve since 50 years. So one of the problems, so here you have the example of a SEEG recording of an exemplary patient. So the seizures start here in the prefrontal lobe, and then it will propagate to the premotor area and the cingulate cortex, and the patient will start to have an altered behavior. So it will start to cough, to touch the nose, cross the legs, and the seizure will continue to propagate. Here in the temporal lobe, it continues to propagate in the premotor area, and the patient will start to singing and tap in the nurse's hands. So as you can see, epilepsy is not restrained to one single area in the brain, but involved a large network, and it's one of the problems of epilepsy. So if some surgery are not successful, it might be because the clinician did not identify all the area involved and the pathway in these networks. So one of the questions will be, can TVB predict or refine better these epileptogenic networks? So I will present you now how to use TVB to model this epilepsy spread and how to build a virtual epileptic patients. Here it is a standard workflow for the clinical applications. So first, we get the imaging data of the patients, so the structural data, MRI, and the DTI to reconstruct the anatomy of the brain. And once we have this anatomy, as for TVB, we choose an appropriate model to reconstruct the functional activity of the brain. And then we have a personalized functional brain network. With the clinical hypothesis, for example, in the case of the epilepsy, which regions are the epileptogenic zone or the propagation zone, we can refine the network in this brain model. And with the simulations and the empirical data, we can fit our model and give some indications to the clinicians for a better prediction or better identification of the network. I will present you the examples that was published. So the virtual epileptic patient is a paper from Victor Gilsa in 2017. So in this paper, they use the data of one patient who have two types of epilepsy. So one type, in this one, is the simple seizures. So the seizures start here, in the right hemisphere, in these electrodes, and stop. And then the patient also exhibits complex seizures. So here again, the seizure starts in the right hemisphere, but this time it's propagated to the other hemisphere, here. And here it's an example of stimulations in the brain of these patients. And if we stimulate these regions, we see that it propagates, like for here. So just a reminder of the equation of the brain network model. So here, the big G and the small G came from the personalized data of the patients. It is the structural connectome, either at the here if we are using a surface simulation, and here if we are using a region simulations. Here we have the Neuromas Model that we have to identify to reproduce the epileptic activity. So as I say, for the workflow, and as you should know now, the first step is to bring the individual data of the patient inside TVB. In particular, the structural connectivity matrix. And then we have to choose a specific neural mass model. In the case of epilepsy, so there is a specific model that was developed in Marseille. It's Epileptor. And this model was developed based on the identifications of the bifurcations at the onset of the seizures and bifurcation at the offset of the seizures. So you will have an in-depth talk about the bifurcation theory tomorrow, and an example how to draw your first bifurcation diagram. But just to understand it, your goal is to reproduce a biophysical signature patterns. Here it's the fast discharges. And in the field of the dynamical system, we have this type of bifurcations where we pass from one state to another. So here we pass from a stable state to a solitary state. So we have these fast discharges. In the epileptor model, so there is different type of bifurcations going from rest to limit cycle or from limit cycle to rest. There is a, as we say, a classification of taxonomy of seizures. 80% of the seizures across the patients are saddle node and saddle homoclinic bifurcations. So they built and they developed the epileptor model based on this type of bifurcations. So the epileptor model is phenomenologic, so the variables inside the system are not linked to a variable biologic, but the global behavior of the system will reproduce this, will reproduce realistically the behavior of these systems. So epileptor is composed of 5 state variables. The first populations, here, these first systems reproduce the seizures activity. So this low voltage fast discharges. But we have a second generic pattern in epilepsy. It is a spike wave events or interictal spikes that we have seen in the first slides. And it is these second populations that allow to reproduce these spike and wave events. But in epilepsy, you have 2 states, the ictal state, so the actual state of the seizures, and the interictal, where the patient is in a normal state. So we need the system to switch autonomously to one state to another. And it is the role of these variables that will allow the system to switch between here the interictal state to the ictal states. Okay. And one important parameter in the model is the variable x0, who can determine if my system is trigger a seizure or it is in a resting state. So when this variable is below a certain threshold, I'm in a fixed point. But when I'm crossing this threshold, I can trigger a seizure. So we say that that the system it is in epileptogenic. Okay. And the output of the epileptor, it is a combination of so here we can x2 minus x1, which means it is the output of the fast of the system's x1 here, so the fast discharge, and the x2. So now we know how to model one node to be epileptogenic, trigger seizures, but we need to link with its neighbors to build a brain network. And here we insert this the coupling function in these slow variables, and here C, it is your structural connectivity of the patients. And the fact that we are introducing the coupling on the slow variables, it's because it was shown in the paper of, it's not written here, but it was another study that shows that why we put this coupling on the slow variables and not on the fast. It's to reproduce this propagation pattern that we observe in the experimental data. So once we have the brain of our patients, we have choose a Neuromas model to reproduce the epileptic activity. We can refine the network of the patient by including the clinical hypothesis found by during the pre-surgical evaluations. So for this the example of the patients, she had hypothalamic hamartoma, which is it's a neural mass attached to the hypothalamus. And to introduce this anomaly inside the brain network, we will add here the local in the equations. So first, we will define this anomaly as a new node in my networks, and then to add the influence of this anomaly, we will add a local connectivity in the equation of the nodes. A second hypothesis can be on the epileptogenic networks. So from the clinicians determine that in the red zone, it is the epileptogenic zone, so where start the seizures. And in blue, it's where the seizures spread. It is an example where the seizures start and then propagate in the network. And for each regions, we have a different type of excitability. And this different type of excitability will allow us to build this propagation pattern we observe in the patients. So we will build a heterogeneous brain. Each brain area will have a different set of parameters. So once we have input in TVB these three main components, we can simulate the behavior of the patients. So in TVB, we have the possibility to simulate neuroimaging data, and in particular the SEEG. We have a SEEG monitor inside TVB. And then you can compare your simulations with the clinician prediction. So here you have on the left, so in yellow, so here in the yellow zone, it is the epileptogenic zone, and in red, so here it is the level of the excitability of the node, and in red it is the epileptogenic, the propagation zone. Here it is our simulations, and as you can see, it is there is some similarities between. And here are the simulation results for these patients, and they were able to reproduce the simple seizure, so starting in the right hemisphere here and that stop here. Reproduce the complex seizures that start also in the right hemisphere and then propagate in the left hemisphere, or even reproduce the stimulation case. So this virtual epileptic patient is able, so as I said, able to reproduce both behavior of the patients. Now I will show you some use case, so different studies that were done using the epileptor. The first one, so they used a linear stability analysis. So they used the connectome of the patients and the hypothesis about the epileptogenic zone. And computing a linear stability analysis, they were able to predict in which direction the seizures propagate, so identify the propagation zone. Here there is a clinician prediction, and here you have the predictions using this linear stability analysis and the epileptor model. So here again in yellow you have the epileptogenic zone, and in red you have the propagation zone, and you see that there is some discrepancy between both. So the LSE found another regions in the propagation zone. So they validate this study across, the first study here was done with 15 patients and they extend to 50 patients. So here you have the score of the predictions of this propagation zone. If we are using individual connectome, if we are using the connectome of control, or if we are using connectome shuffled. And we have a very high predictions when we are using the connectome of individual patients. Here, they compare the number of regions, the discrepancy with TVB, to the angle score, so the output of the surgery. And we see that the more TVB found discrepant, so when TVB more the discrepancies is higher and more I have a worse angle score. A second study is to, okay, first we have our epileptogenic zone, we have identified the propagation zone. But what we want is to stop the propagations, that the patients doesn't have these altered behaviors. So if we are going back to an example of a patient, this patient exhibits, as in the previous case, here, seizure start in the yellow zone and propagate just here, and the other one is propagate to a larger zone. So what we want is to stop this propagation, so cut this connection. So either we, as in the surgery, we remove completely the epileptogenic zone, or we can also just cut the link between the nodes with gamma knife or laser surgery. Here we have 15 patients, the same 15 patients that in the previous paper used. And here you have the number of lesions. So in black, it is the number of lesions when we remove completely the epileptogenic zone. And in green here, it is selected lesions, so we identify the links to cut. And we see that when we identify the link to cut between the nodes, we have a minimally invasive resections. The third use case is the Bayesian virtual epileptic patients. So here they will use the SEEG data of the patients and they will compute the power spectrum and the energy of the time series in order to perform data fitting and in particular machine learning. So the invariance variational bias. So they will identify a distributions of parameters for each regions and which region can be epileptogenic or propagation or LC. So this is an example. So here we have it is a parcellation of the brain of 84 regions, and for each regions, they compute a distributions of these excitability parameters. When we are, so -2 here, it is the threshold we identified earlier. So when we are below this threshold, we cannot trigger seizures. So here, IZ, so who have a very low excitability, it is the nodes, the regions not involved in the seizures, the healthy regions. When we are above the threshold, the regions trigger autonomously seizures. It is an epileptogenic zone. And when we are close to this threshold, it is a provocation zone. So when these nodes are, we can say, healthy, but because they are in the networks and links to this node, this seizure will propagate here and push the node above the threshold to trigger seizures and turn the seizures. So in France, so in particular in Marseille, they launched a very huge clinical trial where they will receive more than 400 data from patients. They will build these virtual patients, apply this machine learning technique, fit with the SEEG data of the patient, and identify. So this is an example of the clinical report for these exemplary patients, and identify which nodes are from the epileptogenic zone, the propagation LC, and so there is, in particular, the epileptogenic zone that is the target of the epilepsy surgery. So they will provide a clinical report to the surgeon, and help them to take an informed decisions. So some clinic will receive back this clinical report, others not, and they will identify if. So of course, the clinicians are free to take into account this information or not, but the goal is to see if we are increasing the success of surgery by adding this information of TVB. And there is a last case. So for the previous case, we were studying during the seizures, the activity, and in particular using SEEG data. And SEEG data is in invasive techniques. But we have, and also, seizures are very, we cannot predict seizures, and from one patient to another, some patient can have only one seizure per week or per month. So it is very difficult to record such seizures, such patients. In particular, the patients in Marseille are recorded during 14 days in the hospitals. So they have this implanted electrode during 14 days. They are recorded during these 14 days, but if they have, of course, only one per week, they will record very few seizures, and it's difficult after to identify the epileptogenic zone with a certain, to identify which regions. So one possible alternative will be to use the data when the patient do not have seizures, so during the intellectual state. And we have a lot of this data, and one of the technique to identify brain activity during resting state is the fMRI ball data. So the epileptor at the basis was developed to reproduce these epileptic seizures, but it was developed to identify the type of seizures and reproduce the epileptic activity. But outside the seizure, during the intellectual state, epileptor is in a fixed point. And when we had the noise, so it will be, the resting state will be just noise. But we know from a lot of studies that resting state is not just noise. We have some structure inside. So we need to put the system in a certain configurations that is able to reproduce this resting state. So a combination of noise and of oscillations. To do this, so one of the system, so in TVB, if we are putting the system in such configurations, we will lose one of the marker of the interictal state. It is the interictal spikes. So we cannot do this with the epileptor. We need to extend it and add another populations. And it was done here by adding the third populations with the OP subsystems. So this system was really used to reproduce the resting state in resting state network activity in previous literature. And so now the output of this extended epileptor will be not only the combinations between, so not only the output of epileptor, but also combined with the output of this OP model. And we have had these parameters to scale the epileptic activity inside the systems. And so we can reproduce three types of activity, either resting state, or resting state with interictal spikes, or resting state with seizures. So the first step of the study was to compare, to find. So we are looking first and comparing with healthy control subject. So we are using connectome from control subject and connectome from the same 15 patients than before. We put into our system, and we look where is the working point of the system. So the working point, it's where my model fits the empirical data. And we are looking with two different parameters. One is the excitability of the systems, and the other one is the coupling with the remote region in the brain. And it was shown that there is no difference with the coupling parameters, but for the parameters for the excitability of the node, it was shown that there is a shift toward lower excitability in the patients, which is surprising because we will have suppose that they are more excitable. So then we wanted to see if we had the interictal discharge inside the model, if the functional, yes, here we are looking, I forget, to fit the empirical data with the simulated data, we compute the functional connectivity, either dynamic, so there were 5 metrics. One is the static functional connectivity, so the basic ones. Other one is the dynamic FC. There is also the metastability, or even the multiscale entropy that you have saw yesterday. So here they perform an exploration to see if by adding interictal discharge, the functional connectivity will be altered. So here you have the regions ordered by node strength, anatomical node strength. And here we have used a parameter that allows to increase or decrease the spike frequency in the model. Here I just choose to just have a look to the simplest functions, the correlation functions. Here we looked if I'm adding only one spiking region, if I will induce alteration in my network. And here we see that it is the same everywhere. So one region in the network, one spiking region will not involve or alter the whole brain networks. And here they create, they reproduce the spiking network of the patients based on the epileptogenic network identified by the clinicians. And it is ordered by some patients have only two regions in their epileptogenic network, other 9, and we see that it doesn't change a lot neither. I didn't show the figures, but they did another explorations. It's okay. Only the excitability, the global excitability of the brain is changing in the epileptic patients. The interictal do not change the overall connectivity. But in the literature, we know that between the control subject or even control regions and pathologic regions, we have alterations in the connectivity. So they modify the excitability of the epileptogenic node only, and see if my nodes are more excitable, I will increase or alter the functional connectivity, the overall connectivity, and also fit better to the empirical data, and it was shown that yes. So it is, yes, the overall study for the epilepsy. And now we will try to reproduce a virtual epileptic patients, but with the Jupyter notebook.